Thank you everyone for giving me the opportunity to present EyeStem at the World Stem Cell Therapy Summit. It's a very important event for us, and we're honored to have this privilege. My name is Dr. Jogin Desai. I am the founder and CEO of EyeStem, and I will take you through the next few slides to talk about what EyeStem is about, what we're trying to do, and how we're looking to change the paradigm of cell therapy globally. I will go off video so that you don't see this annoying thing everywhere in the slides. But if I go through the slides, what do we do, and what's the problem that we're trying to solve, right? We'll take you through these 8 things that I have on my agenda. So the biggest problem that we have, as you see, is that current cell and gene therapy products for incurable diseases, all of them are estimated to cost in excess of $250,000 per injection. The lowest estimates are coming in that, but everything is 6 figures in dollars, right? This is untenable. It does not make any sense, especially for the developed world, but I would argue also for the developed world. So we're looking at presently resolving this problem in 3 conditions. Starting with the first one, which is dry AMD. About 170 million people in the world suffer from it, 40 million right here in India. There's no cure for it. As everybody knows, wet AMD is treatable by drugs like Lucentis and all of that, and that's global market value for that is about $8 billion. Dry AMD is about 9 times that of wet AMD as per incidence. So it's a very, very, very big problem. The second one that we're trying to treat is retinitis pigmentosa. Dry AMD happens in patients above 50 years old. RP is the exact opposite. It happens in children, and we're looking to treat that with photoreceptors. I'm going to introduce the assets that are going to treat each of these diseases in a little bit of time. And the third one is IPF. In some ways, it's even worse than cancer because once you're diagnosed, you have about 4 years to live. So these are the 3 that we have gone after, and our mission is to democratize access to cell therapy and provide benefits to a larger section of humanity. It only when we get therapy costs to a point where what I call the bottom 99.99% can take this, then only will cell and gene therapy become meaningful as in terms of their impact, and that's what we hope to do. We also want to make sure that we set global benchmarks for quality, because quality is a given in this space when we do anything that is given to patients. And we want to use our cost leadership to establish ourselves as a globally consequential cell therapy. I'm going to take you through what our assets are now. First is EyeSight RP. It is a suspension of retinal pigment epithelial cells. Our first in human trials are going to start very soon, and as a philosophy, once again, if you go back to our basic mission, which is democratize access, for that you've got to have a cost which is not just 10% lower than competition, but in my view 90% lower, because that's the only way it will get to a point where it can actually be affordable to most people. So which is why we went with a suspension of cells. We went with an allogenic therapy, and we started with the eye, because the eye obviously immune privilege and all that sort of stuff, but it also has, you need a lot less cells in the eye, like 100,000, 200,000 cells, and you're done. And our product is frozen so that it can be stored. We've got some fantastic stability data and so on and so forth. So we're very excited about this. And one of the reasons why we are excited is our partners at OHSU have reported all this. All the data that you see here is all from OHSU, all the text. And we're very happy about it because it provided significant, obviously I will not read everything on the text, but there was a significant beneficial effect in the RCS rat. There was no safety concern that they saw. And as per the scientists of OHSU, as most people know, a lot of these macular degeneration studies get done there. It's a very promising treatment that could match or exceed competitor cell products. We have a patent in India and a patent in the US on the process that we have. And I'll talk a little bit about the process as I go along. Here's our regulatory roadmap. Every product for us will go through the same regulatory roadmap. So we'll go through first, do we have a protocol for a consistent quality product? Can we show it in a in a dish that, for example, for RPE, there is PEDF and BEGF and all that is upregulated and so on. Can we do that? Can we show that there are no iPSCs in there, or they are below the level of detection? Then we did a pilot animal efficacy. Do we publish in peer-reviewed journals? All our publications are on our website, EyeStem.com. We finished our CMC. We chose to do pre-IND with the FDA a little bit earlier than usual, because there is a lot of, this is a new therapy, and we wanted to make sure that before we move further, we have all the clarifications that are required from the US FDA, because and that includes everything from which animal model to use for tumors, which animal models to use for efficacy, how many rats to use, how do you validate the device, how do you, what is the clinical trial outline, how are you going to dose the patients, are you going to use a placebo, so on and so forth. Every single thing we've got. So we've got some fantastic feedback from the US FDA for this regard. We finished our assay validation, our GMP protocol, and we did a dose ranging efficacy at OHSU as well. We've got now two things that are going on, which are all three things actually, which are all going to culminate by end of September. The subretinal tumorigenicity in RNU rats. We have a GLP tox that is going on, and with the same batches, we're going to do a final efficacy at OHSU. So we're looking at filing in India in November of 2022. We're also looking at Australia as the next market to go to once we get the first signal of the product being effective and safe. Here, just a copy of the patent that we have. The next product is eyesight PRP, and again, same philosophy, same approach, allogenic, frozen, and addressing a site which is small enough that you can achieve scale. Right? We've got again the patent, and the good thing about a patent is it is a unified protocol. So you can actually develop RPE and photoreceptors through the exact same protocol. In fact, our patent is for retinal pigment epithelium, photoreceptors, and retinal organoids, all through a process that starts similarly and then sort of diverges as we go along. We're very excited about that. This one as well, because the impact will be significantly higher. A little bit less ticks on here than the previous one, the check marks, because we're looking at this 1 year behind. But since the beauty of this is we have a unified protocol, so the reagents that we have, so the pathways that we have, the quality checks that we do in the GMP side, and they are 80% similar. So your CMC becomes much easier. Your assay validation becomes much easier. The only thing that you have to do is, do the final few assays for the matured photoreceptors. That's the only one that you would need. Right? Again, talking about the patent, and we're talking about idiopathic pulmonary fibrosis, which is where we are the leading player in the field. We've got some very promising animal data, which shows that we should be able to decrease the collagen in the rat, and so that is because that's what drives the pulmonary fibrosis. This is where things get a little bit complicated because you need a lot of cells. So we're figuring out how to create an expansion strategy around these cells to make sure that it is on the right track. So again, could have a substantial impact. We're looking at some immune privilege strategies as well, including HLA null cells, which we have a license for. Because the lung is not as immune privileged as the eye, so there is a significant problem that one would face when we inject cells. So we're figuring all that out as we go along, right? Again, a similar slide deck. I won't go into each one of these, but less checks than the previous ones. But still, we've got to a point where we understand the pathophysiology much better and how the cells should interact in order to correct the pathophysiology. So we're fairly optimistic about this. About the science, here's how these stem cells transform to RPE tissue. We've got the quality checks along day 30, day 40. Starting at day 18 and so on, and these consistent quality checks lead to a consistent quality product. This is how it looks. And this is why we feel we are optimistic about the cell suspensions, because each tube can treat 20 patients. It's true, has 5 million cells. We've got stability data of up to 3 months. By the time we get to the clinic, we will have stability data of up to 9 months. And we've got a very intense product characterization to make sure that we've got no iPSCs in it. We've done spiking experiments to make sure that it is very safe, and so on and so forth. So here's a quick look at the EyeStem platform. Right, first thing is the self-reading cell sources. We acquired clinical grade iPSCs from NIH, and we've got a very good agreement with them. This is the same lines that are distributed through RUCDR. We're following a very well-established path here. We didn't want to reinvent the wheel. We also have a license from IPSHA to make sure that there is no risk for this important source. This is the core iCellDiff protocol. This is the secret sauce, if you will, of EyeStem. Which is a proprietary, efficient, repeatable, and predictable process for generating target cells from clinical grade iPSCs. Each one of them, we have to take an input, or apply a particular number of factors to it, and create a final product. There are some critical gates along the way, and so on. But the same philosophy applies, and we have already commenced active GMP production now. For we have a master cell bank in India, and we have commenced GMP production of the RPE product. And we can increase the scale of the process by increasing the size of the culture vessel and all of that. But we will look at that once we get through the first in-human data. Right? Now, these are our partnerships. NIH access to the clinical grade lines. We've got the best ophthalmology institute in India, which hospital in India, where we are going to do GMP manufacturing as well as do first in human trials. Aura is based in Boston. Helped us with our pre-IND. We've got a global license for HLA null technology from Pancella. OHSU is, of course, our animal study partners, and IPS AGA, from which we licensed all the iPSC patents. And then finally, of course, trained personnel. Where would you go without that? We're very selective in doing this. Each recruit undergoes training. There are 2 labs, one which everybody can enter, the other lab where only a few selected people can enter. And this, again, is part of the secret sauce. Much harder to define, and much harder to execute, as everybody knows. Right? What do we want to do? First, we want to focus on incurable diseases. Then, secondly, once we decide the disease, we would do research in-house, develop protocols, whether we can apply the iCellDiff paradigm to this target tissue. Once we do that, then we move this out to third parties, whether it is preclinical, regulatory, or clinical studies. And then we go to first in human in India at low cost, and then develop cost down to reduce the cost even further. The assumption, the hope is that we will have a reverse integration every single time we develop a product, so that we can capture the institutional experience and then keep reducing the cost further and further. Right? Our growth strategy is very simple. There are 2 things that we want to concentrate on. One is there any automation that one can provide as we go along, and is there any artificial intelligence that we can put on top to make the product more repeatable, the process more repeatable. We're currently in talks with several early-stage pharma companies, and ultimately, as soon as we get first in human data, we will start these combinations. A quick one about the structure. I myself have over 20 years of drug development experience. I was part of the team that set up Quintiles in India. I was heading up the global cardiac safety business. And then I became the CEO of a joint venture known as Senduit, which was a joint venture between Quintiles and Thermo Fisher in North Carolina. And so I have significant experience working with the US FDA, EMEA, MHRA, and so on. Dr. Bhattu is a retina surgeon and the chief medical officer. And we've got Dhruv, who is right now the founding director at the core lab in Cedars-Sinai. So we've got a very balanced management team in front. And Dr. Paul, of course, who is the main guy behind the iCellDiff protocol. Fantastic advisory board. A lot of people you would know on here. Dr. Rao, Dr. Bharti. Dr. Bharti is obviously running an RPE trial in the NEI, and Dr. Rao was the founder director. So we're very blessed that we have a fantastic advisory board who helps us make a lot of crucial decisions. These are our pre-Series A investors. I, of course, invested in the company, but these are all former Quintiles people who Appel was the president of the global clinical development at Quintiles. Gert is a prolific biotech investor. We are very fortunate. We got two institutional investors from India. Kotak is the largest private equity group in healthcare in India, and India Partners is one of the most sophisticated healthcare investors in India. So we're very happy that we've got it, and we're right now closing our Series A round. There are some legal reasons why we can't disclose all the Series A investors coming in, but there's good, we're very happy we've closed our Series A fund. We're close to closing our Series A funding as well, which is great. Let me stop this by giving you a little bit of the India perspective and what I've seen, because my experience has been there are some organizations that I've run in the US. There are some that I've done in India, but they've always been global. So it gives me a good perspective on what works in India and what doesn't work in India. So the pros and cons, right? First of all, huge addressable market. Unfortunately, right? Even the rare diseases in India are not rare, unfortunately. And there are 1.5 million kids with RP in India. And I'm sure if you look at IPF and so on, but it will be more and more. The middle class has very good buying power. I don't think this is appreciated as much because India is moving up. It's the 5th largest economy in the world. About 600 to 700 million people have buying power, which is bigger than most Europe. So there's a massive market to be tapped. We got excellent scientific manpower, unfortunately available at a very low cost, because science typically is only, it's not seen as a career just like IT, for example. Science is only people who are really passionate about science go in. We're hoping to change that at EyeStem and make sure that the wealth creation is also part of being a scientist. Just because you're a scientist does not necessarily mean that you should not make money. And we've got some fantastic biotech incubators where there is excellent plug and play facilities. There's lots of people who've come in from abroad, including Dhruv, and said, the kind of infrastructure you have available rivals anything that they have seen in the West. So, so that's a great thing. On supply chain issues, it hits us much harder because it takes a long time. It takes about 6 weeks for me to order a reagent. So you have to plan in advance, and customs duties in India are still higher. So in fact, a lot of people say doing research is less costly in India. I would say sometimes if you add the time factor as well as the cost factor of the reagents, it might be more expensive, at least to do R&D. The regulatory roadmap is uncertain. That is obvious. The ecosystem for cell and gene therapy is not really developed here. There are very few players, and that's why this is expected. We've been talking to the regulators, and they've been very supportive of us so far, and they've been, going out of their way to help us to move this forward. The total science infrastructure is significantly worse. If you wanted to do, for example, if you wanted to do an RCS rat model here, it would not be possible because the people who can do subretinal injections in rats are far and few around. Right, the test, optokinetic tests, and all of that stuff. It's not really easy to do that. So we end up doing all that at OHS. And venture funding for biotechs, even Series A, I would say, is almost non-existent. There is no prominent player who would invest, who would understand the intricacies and the nuances of a business which is into cell and gene therapy, and that creates problems. We're trying to solve them. As I said, we're optimistic, and once our Series A is announced publicly, we will be able to provide a strategy which we believe is unique for us to going forward. But all in all, it's a very exciting time for EyeStem. We are on a mission to democratize access to cell therapy worldwide, and we believe with our first product we will make a significant mark, and hopefully create a virtuous cycle of research and development through which a lot of such products come down the line over the next 10-year period. So I want to thank the World Stem Cell Therapy Summit once again for providing me the opportunity to share this information. And if you have any questions, my email ID is on the slide, and please do reach out if you need. Thank you very much.